are going to discuss about the metabolism. Now always remember what all points we have to focus. Metabolism revolves around two things. One is anabolism. Anabolism is synthesis of something. Second is catabolism. Catabolism is degradation of something. So whatever energy is required for the synthesis of something, that energy is given out during the breakdown of something. So the energy, the flux of energy remains the same, and that is what first law of thermodynamics also says, that energy can only be converted from one form to another, right? So now see, again, the most important thing here is, if we talk about carbs, the principally important monosaccharides are glucose, galactose and fructose. Since they are highly soluble in water, the first thing which occurs is phosphorylation of this. Phosphorylation means we attach a phosphate in them. So obviously if they are with a charge, they cannot move backward. So because if they move backward, they are going to disturb the entire osmolality. So this becomes one question also, that what is the mode of phosphorylation? Yeah, what are the enzymes required for phosphorylation of this? Right. Now how we are going to study? We will be studying the major pathways. Major pathways means whatever carbs we are digesting, what is the ultimate destination of glucose. And then we are going to study the alternative pathways. Major pathways, the most important one is glycolysis. Now see, glycolysis is there, glycogenesis is there, TCA is there. Today we are going to focus mainly on glycolysis. Alternative pathways, the major pathway is, you can say alternative is hexose monophosphate shunt, and the other one is uronic acid pathway. Right? So today we will be discussing about glycolysis. Now, what you have to remember in the processes. First of all, you should know the site. Site means where it occurs. Second is, you should know what kind of pathway it is, whether it is anabolic, whether it is catabolic. Then you should know about the energetics. Energetics means how much amount of energy is given out or how much energy is taken. That will come under energetics, and the most important point is regulation. Now see, coming over to the site of glycolysis, glycolysis occurs in the cytoplasm. Then what kind of process it is? It is a catabolic process, and it occurs in both the mediums, means it occurs in aerobic medium and it occurs in the anaerobic medium. Means it can act in the absence of oxygen, it can act in the presence of oxygen also. Coming over to the energetics, aerobically it produces 7 ATP. And if we talk about anaerobically, glycolysis produces 2 ATP. Again, a very important question asked in the exam. Question is asked like this: one molecule of glucose via aerobic mode of glycolysis produces how many ATP molecules? So the answer will be aerobically it is 7, anaerobically it is 2. Then comes a regulation. Now this glycolysis is a 10 enzyme catalyzed base reaction in which 3 reactions are strictly irreversible. 3 reactions are irreversible, which are: first reaction is glucose to glucose 6-phosphate. Here the enzyme is hexokinase. Second reaction is conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. The enzyme is PFK. It is also written as PFK1. And the third step is conversion of phosphoenol pyruvic acid to pyruvic acid. Right? Here the enzyme is pyruvate kinase. So obviously, if these are irreversible reaction, I can easily say that they can be the regulatory ones, because you know the reaction which is irreversible can easily regulate. So again, if we talk about the mode of regulation, glucose to glucose 6-phosphate is simply feedback inhibition, or it is a rate limiting enzyme. If we talk about fructose 6-phosphate to fructose 1,6-bisphosphate, it is rate limiting as well as a rate committed enzyme also. This I discussed again in detail yesterday, because this question has been asked so many times in the exam and always there is a confusion. But still, again I will be rectifying the confusion here itself. And the third is pep2 pyruvic acid. It is again a rate limiting enzyme. Right. Now coming over to the definition of glycolysis. Always remember, glycolysis is defined as the incomplete oxidation or breakdown of glucose. It is always incomplete, means we cannot say कि one molecule of glucose is completely broken down by glycolysis. Then one molecule of glucose which has 6 carbon atoms, by incomplete breakdown it forms 2 molecules of pyruvic acid which has 3 carbon atoms. That is why it is an incomplete breakdown. Right. Then third important point is there exists a Rapoport-Lubring shunt also in this Rapoport-Lubring shunt, in which only 10% of glucose enters. Right. So these are some of the important points, and the fourth important point is, if we talk about the hormonal regulation, we can say that insulin, this is very important, insulin always activates glycolysis, and this is an exception also because insulin is an anabolic hormone, but this is the only catabolic process which is accelerated by insulin. So insulin accelerates and glucagon inhibits glycolysis. Right. Then moving on to the reaction. By this just we can say all the reactions in glycolysis are reversible except 3. Now we will discuss the first irreversible reaction in detail because the question has been asked on that. What happens is this glucose is converted into glucose 6-phosphate. The enzyme can be hexokinase, it can be glucokinase also, and one molecule of ATP is utilized. Right? So we can say that one molecule of ATP is consumed. And hexokinase and glucokinase, the major difference is this glucokinase is nothing but it is simple hexokinase 4. Is glucokinase. This is present in all tissues. If we talk about glucokinase, it is present only in the liver, right? Second important point is hexokinase has less Km, more affinity, whereas glucokinase has more Km, less affinity. And insulin always acts on hexokinase, it never acts on hexokinase. Only hexokinase 4 or glucokinase is acted upon by insulin. And this glucokinase will work in the body only when the concentration of glucose exceeds 100 milligram per dl. So once this glucose 6-phosphate is produced, second step is simple, interconversion, and it is converted to fructose 6-phosphate. Fructose 6-phosphate, the enzyme is phosphoglucomutase, or it can be isomerase also. Then this fructose 6-phosphate by another irreversible reaction forms fructose 1,6-bisphosphate. And here the enzyme is PFK1, and the reason why I am writing this is because this is considered to be the bottleneck of glycolysis. This fructose 6-phosphate, on the other hand, by a reversible reaction is converted into fructose 2,6-bisphosphate. And here the enzyme is PFK2, and reversible is fructose 2,6-bisphosphatase. Right? Then this fructose 1,6-bisphosphate undergoes splitting, forming 3-phosphoglyceraldehyde and dihydroxyacetone phosphate in the presence of enzyme aldolase. And these two are also linked as isomers with respect to one another. Clear? So here we complete one step of glycolysis, which is energy investment step or phase. And we can say that 2 ATP molecules are generated here. Generated in the sense that they are given, means they are consumed. This 3-PGA will now have to, because this is a catabolic process, that means the body requires energy, isn't it? So this 3-PGA will now give energy to the body, because whatever ATP is invested, it will be regenerated back. So this 3-PGA will undergo 2 steps at the same time, forming 1,3-bisphosphoglyceric acid. Enzyme is simple dehydrogenase, and NAD will be converted into NADH plus H positive. And we get 1,3-bisphosphoglyceric acid, and this 1,3-bisphosphoglyceric acid again will form 3-phosphoglyceric acid, synthesizing 1 ATP. Now this reaction is important. Why it is important? Because this is an example of substrate level phosphorylation. First step in which ATP is generated. Now see, ATP is coming from where? ATP is coming at the expense of the substrate, and this is substrate level phosphorylation. Very, very important question which is asked in the exam. This is the first step of glycolysis, which represents this. Then coming over to the next step, this 3-PGA will be converted into 2-PGA. This 2-PGA will be converted into PEP, and finally this PEP will be converted into pyruvic acid, again synthesizing ATP. So this is the second step of substrate level phosphorylation. So you can say that if we have to summarize, there are 2 steps which are an example of substrate level phosphorylation. Alright, this is what is glycolysis. Now, one point I told you about this is, you can say this glucose, we started with 1 molecule, and we see that it ends with 2 molecules of pyruvic acid, which has 3 carbon atoms. So it is incomplete breakdown. Okay? Then coming over to the energetics, if we talk about the energetics, you can simply say that one is aerobic in the presence of oxygen and the other is anaerobic. Anaerobic is in the absence of oxygen, right? So if we talk about aerobic, you can say that 7 ATP will be produced, because if we talk about this 2 NAD, which are giving 5 ATP molecules, and 2 plus 2, 4 ATP. So you can say 5 plus 4, 9, 2 ATP are consumed, so it will be 7 ATP by aerobic medium. But if we talk about anaerobic medium, anaerobic mein you have to subtract this, so it will be 4 minus 2 equivalent to 2 ATP. Very important again. So this is the energetics. Okay. Then I discussed about a very important topic in this, Rapoport Lubring shunt. See. सबसे पहले तो you should know how do we define a shunt. Shunt is that which does not have an end product. जैसे pathways are there, pathways में always end product will be there, but in the shunt there is no end product. So what happens is, in RBCs, in our red blood cells, a ratio of ATP to ADP is inactive. Means this step can be bypassed in RBCs because it is not required. So how it is bypassed? This 1,3-BPG will be converted into 2,3-BPG or 2,3-bisphosphoglyceric acid, which will be converted into 3-PGA. Means in the presence of simple inorganic phosphate, hydrolysis will be there, and what we get is 2-PGA. So you can say glycolysis continues. Now the question arises, which might strike your mind, is what is the significance of this 2,3-BPG? The significance is it plays a very important role in hypoxia. In hypoxia, the level of oxygen decreases. So what happens is this RLS will increase. RLS is Rapoport Lubring shunt, it will increase. So this 2,3-BPG, the concentration increases and it unloads maximum oxygen to the tissues. So obviously if it is unloading maximum oxygen to the tissues, our body can survive these hypoxic or anoxic episodes. You got my point? Simple is there कि approximately 10% of glucose enters this RLS. ज़रूरत क्या पड़ी? Why it is going into RLS? Because there is no need of requirement of this ATP to ADP ratio. So this step can be bypassed. And how this step is bypassed? Simply because phosphate to we have to remove, because glycolysis is going on. So 1,3-BPG will be transformed to form 2,3-BPG, which is converted into 3-PGA. अब question जो आया है, the question has been asked about the net gain of ATP. So if we talk about the net gain of ATP via RLS, it will be 0. Net gain of ATP via RLS will be 0. Why it will be 0? Because this does not stand. So it is 2 ATP are produced minus 2 ATP equals 0. So you can say that RLS होती तो है, but RLS से no ATP is synthesized. But फिर भी क्यों होती है? Because it is helpful. It plays a very important role during the hypoxic conditions because it is unloading more and more oxygen to the tissues. Okay? This is what is glycolysis, the energetics. Clear? And there are two reactions which it is clear also, two reactions of substrate level phosphorylation. One is 1,3-BPG to 3-phosphoglyceric acid, and second is pep-2 pyruvic acid. Right? So this is what is glycolysis. Then another important point about glycolysis is energetics we have discussed, site we have discussed, it is incomplete breakdown, that also we have discussed. There are the regulation if we talk about. So regulation may always remember hexokinase has a rate limiting condition, and the second PFK1, this is very, very important. PFK1 is a rate limiting enzyme as well as a rate committed enzyme. Rest both the enzymes are simply rate limiting. Right?